All right. Good morning from Michigan, or good afternoon, or good evening, or wherever you are, depending on wherever you are. And welcome to the ZOOMinar. Today we have two early career researchers, Dr. Theillet from Paris and Dr. Tae Hun Kim from Toronto. The first speaker is Dr. Francois Theillet. He received a PhD degree in molecular biophysics from the Pasteur Institute in Paris in 2009. Then he was a postdoctoral fellow in Philippe Selenko's lab in Berlin, pursuing in-cell NMR research. After completing the postdoctoral research, he joined the CNR Institute of Integrative Biology of Cell in Paris-Saclay in 2016. As an independent researcher, his lab is investigating intrinsically disordered proteins and kinases that have biological roles in cancer and neurodegenerative diseases. So with that brief introduction, Francois, get started.
Okay, thank you. Thank you, and thank you for the invitation. So yes, today I'm gonna focus on the latest results we got in the field of in-cell NMR, and well, let's say I'm gonna show you some of the results we have while trying to improve the experimental conditions and the sensitivity of in-cell NMR. So the questions that we want to tackle are related to these little movies. These are molecular dynamics trying to mimic the cellular conditions, where we see that the macromolecular crowding can be really high, and it can really affect the protein tumbling, macromolecular tumbling. You also have quite large concentrations of metabolites that can really affect the physicochemical properties of your buffer, and so affect the structure, the conformational behavior, and interactions between proteins. And speaking about interaction between proteins, you have, because of this high molecular crowding, a lot of specific or unspecific interactions between macromolecules, and these can also affect the structure and the dynamics of the proteins in general. And these can have consequences not only on structure, but also on interaction between partners, and also interactions between, for example, drugs and their protein targets. And all these questions, where we want to have experimental information on these questions, and the way we try to obtain this information is by performing in-cell NMR experiments. So what's the principle? Basically, we try to detect C13 or N15 labeled species in a cellular background, which is made mostly of C12 and 14N molecules. Basically, these C12 and 14N species are, so to say, invisible for solution NMR. And so so if we manage to deliver or to label proteins in cells that are C13 or N15 labeled, we will be able to perform isotope filters, NMR isotope filters, and detect selectively the species of interest. So all the details about what has been achieved in the field in the last 20 years are explained extensively in these two reviews here. The first one in chemical reviews is mostly focused on structural biology, in cell structural biology. And the second one is a little bit more comprehensive because it also explain everything that has been achieved with living cells in general in the spectrometer since the 1950s. So you have everything about the NMR of water, salts, membranes, cell walls, metabolism, etc., in leaf cells. So if you want to have all the technical details or the questions that were tackled in the last 70 years using living material, living cells, and NMR, these are the two papers you want to read. I have, so even though you so you can read the two reviews, but I can also explain you very quickly how we produce in-cell NMR samples. So the first, so 20 years ago, when the in-cell NMR focusing on proteins started somehow, people started to use the basic approach that every structural biology labs know. It's overexpressing proteins in microorganisms like bacteria. So you grow your bacteria in natural abundance media, and just before you induce the expression of the protein of interest, you just change the medium. You put your bacteria in C13 and 15 labeled medium, and after a while, because the overexpression of the protein of interest is so high, you can detect mostly the NMR spectra of this protein of interest in bacteria. If you didn't sonicate before the bacteria, of course. So the second strategy was to purify the protein of interest, like we do every day, most of us, and concentrate at very high concentration the protein of interest and micro-inject it in very large cells like frog oocytes. They are 1 millimeter of large. And the last strategy, well, the last type of cells that we can use is mammalian cells, of course. Those are very often the ones that are the most interesting ones, because we are quite often, I mean, most of our labs are trying to study mammalian proteins. So here you cannot microinject cells one by one because it's millions of cells. So you have to use more advanced techniques like the ones that are listed here. So all of them have their pros and cons. And again, well, if you want to have more details, you can read the two reviews I mentioned. I published this year. Well, the thing is, all these methods are delivery methods. So basically, again, you produce recombinantly your labeled protein, and then you purify it and you deliver it in cells. So, but all these delivery methods have basically similar troubles that I will explain later. But first, I want to give you a few examples of what has been achieved in the last years thanks to these in-cell methods. So the first goal that mostly it's our Japanese colleagues who wanted to show that you could solve the protein structure directly in cells, and they managed to do it not only in bacteria. They did it more than 10 years ago now, but recently also in insect cells. And so these the methods that have been developed are very advanced. Still, I mean, solving a structure using NMR is always a tedious task, and it's a little bit more tedious when it's in cells, because basically the peaks are broader in cells. So they managed to so our Japanese colleagues managed to do it by developing very advanced methods, not only for NMR acquisition but also for processing, and also using very advanced algorithms for solving the structure. I have shown with colleagues some years ago that actually we could also solve structures using pseudo contact shifts by delivering proteins that were attached to lanthanide tags. And this makes it possible to solve the structure by recording only 2D spectra, and in these 2D spectra you can measure your pseudo contact shifts, which are very rich in terms of 3D information, and they make you able to solve the structure using only 2D spectra and not 3D spectra. Still, well, the intracellular concentrations that we have to use are rather high, I must say. But it's this shows you that actually the quality of the structural information we have is rather high, because again it makes you it is possible to solve a structure. So that also tells you that if you had a protein with 2 or 3 different conformations, you could probably investigate the conformational equilibrium using in-cell NMR spectra. Once again, because the quality of information is so high that you can also solve the structure. So today, I'm going to focus mostly on alpha-synuclein and disordered proteins. So I have to remind you that we managed to investigate the conformational behavior of alpha-synuclein in mammalian cells using electroporation as a delivery method. So we produced and purified N15 alpha-synuclein at very high concentration. We mixed it at very high concentration with mammalian cells that were grown in a natural abundance medium. And upon electroporation, extensive washes, and some hours of recovery, we managed to have this kind of situation. So in red, you have synuclein, which is homogeneously dispersed in adherent mammalian cells. And if you overlay the spectra of synuclein in vitro, purified in vitro, and 15 synuclein, so in black and now in red in cells, you see that most of the peaks overlap beautifully, which tells you that the synuclein we detect has the same conformational behavior than the in vitro one. Basically, this is the disordered state of synuclein, which was, and this was the first experimental proof at the atomic scale that a disordered protein could remain disordered in mammalian cells. And of course, we had a few peaks that were lower in intensity. They had the same chemical shifts, but they were lower in intensity. So we measured all the relaxation parameters, and we could show that actually these lower intensities were due to interactions with cellular components that were unidentified at that time. 3 years later, some colleagues in Switzerland have shown that these lower intensities were due to interactions with chaperones, HSP70, HSP90, basically. And so the situation was the following one. We could show that the disordered state of alpha-synuclein was stable in the cytosol, and that we had transient interactions in the vicinity of the phenylalanine 4 and the tyrosine 39, which are the 2 only aromatic amino acids on the N-terminus. We also had interactions on the very acidic C-terminus. And very interestingly, the amyloid region, the central region of synuclein, was fully protected of any interaction with the rest of the cellular components. So that was a long time ago, and there were plenty of, limits in the interpretation of what we did at that time. So not only because the protein was delivered, so we missed all the co-transactional events. They may be important, who knows? And also we were measuring all our spectra at 10 degrees, and I will come back to this problem later. But these are what we could do at that time. So about the troubles, or at least the limits that we have to face with these delivery methods. So first of all, they are very labor intensive because you have to purify very large amounts of proteins and to concentrate them at very high concentration before you mix them and you perform the delivery. So that means not every protein is likely to be studied with these methods because, again, of course, some proteins are stable at very high concentrations, but it's not the majority. And also up to now in the fields, we are limited to low size proteins because of the tumbling, which is even slower in cells than in vitro. And you know all the basic troubles you can meet with folded proteins using solution NMR. And as far as these other proteins are concerned, we were limited to low temperatures, and I will explain you later why. So I wanted to tackle these troubles, and I'm going to show you that we now managed to overpass them to a large extent. And so the basic strategy that I decided to explore was to use in situ expression of the protein of interest. So in a similar fashion to what we do usually with bacteria, you can transfect your mammalian cells. It's just that the promoters, etc., are not the same, but you can have a plasmid, you transfect it. And if you grow your cells in a natural abundance medium and you trigger the expression of your protein in an isotope labeled medium, you will end up with large amount of labeled protein of interest. And maybe you can measure this and detect specifically this protein using NMR isotope filters. So of course you have plenty of pros and cons, but basically the main one is this one, in my opinion. The usual transient transfection protocols don't generate a situation where you have homogeneous populations. I mean, the populations of cells are inhomogeneous. So you see here, if you use cytometry and you have, for example, the expression of a chimera, GFP plus synuclein, you see that among the cell population, you have a very large variety of protein levels. And this is not what you want, because, for example, if you want to study the interaction of a drug with its protein target in cells, where if in your NMR tube you have cells with 1000 micromolar intracellular concentration of the protein of interest, and some of them are at 1 or 10 micromolar, you're gonna report an average, but which is not necessarily meaningful, in my opinion. In the contrary, so we decided to switch to a different strategy. So we generated cell lines with so they are permanently transfected, and they will express the protein of interest only upon exposure to doxycycline. So they are inducible, and this is the type of population we have now. So upon doxycycline exposure, we have a beautiful homogeneous cellular population, and before doxycycline exposure, you see that you have no protein expression. Okay, so what can we do with this kind of thing? So first of all, we tested different homemade media where we supplemented with different isotope labeled amino acids. So it's just a few of them here, and you see that in every case we managed to have the same levels of protein expression. So it's very satisfying, one, because we can have reproducible protein levels. So now, can we detect the protein of interest using N15 or C13 NMR? So this is the basic picture. So we produced alpha-synuclein in cells in a medium containing C13 and 15 lysines. And you see that at the first glance, it's pretty disappointing because before and after, well, with or without, sorry, protein induction, we have basically the same spectra. But if you subtract them, this is what you obtain, and you have a few peaks left here. And if you overlay these peaks, so they are in red here, and you compare the spectrum that you could obtain with uniformly labeled N15 synuclein, you see that you have a number of peaks that really overlap very nicely, and they correspond actually to the lysines of synuclein. We miss some of them, like the lysine 6 or the lysine 10 here, which is very reminiscent of what we observed previously some years ago. These correspond to the fact that we have an interaction on the N-terminus between synuclein and the cellular chaperones. So we lose intensity on the amino acids that are close to the phenylalanine form. And well, one interesting aspect here is that to generate this sample, we just need 10 milligrams of lysine, so that's 10 euros basically, which is pretty satisfying. We are observing here 120 million cells, and the intracellular concentration is 10 micromolar. So this is what you get, and this is another way of showing that synuclein is still disordered in mammalian cells. But here the expression was we expressed the protein in situ, so it was not delivered. It's directly in cells, so we have also the co-translational, the potential co-translational events. We did the same with phenylalanines and tyrosines, and we did also the same with asparagines. And every time you see that we have peaks that are exactly, well, almost exactly at the same place. We have faint chemical shifts perturbations, and we have intensities that are affected by the interactions with the cellular components. So this is satisfying. Also, of course, we miss a lot of peaks, so it's not uniformly labeled here. But there are peaks that we could not observe before, like this asparagine 65. When you have uniform labeling, this is overlapped with other glutamine and glutamate peaks. Here, for example, it's the same here. We have methionines that would overlay with the lysine 45 or lysine 60, for example. So you have less peaks, but at the same time, you have new peaks that you can exploit for So, okay, this is great, but this is still at 10 degrees. And the reason why we have to measure at 10 degrees, it's because you have an exchange between the amide protons and the hydroxide present in your sample. So basically, when you increase the temperature, these other proteins are prone to, well, they have a very fast exchange here on this amide function, and you will lose the intensities for many amino acids. So here it's with synuclein, the model disordered proteins. So you still have a few peaks here, but it's not very satisfying. So some years ago, I decided to switch to a different type of correlation, which is the correlation between C-alpha and carbonyl. So we have a direct carbon detection here, and this is immune to whatever kind of exchange on the backbone. And you see that whatever is the temperature, now we have the same number of peaks, and we have residue specific information, because we have 140 peaks here in this spectrum, and this is also true at higher pH. Okay, so now with this carbon direct detection, are we able to detect our disordered proteins in cells? Yes, well, I just wanted to mention that it's very important to, especially for synuclein, to measure at 37 degrees, because we know that synuclein interacts with lipids, and we have shown, I mean, this is just a quick picture, a quick test that we performed some years ago. So if you take brain polar lipids, you make large unilamellar vesicles, and you measure your NMR intensities, peak by, so residue by residue. At 10 degrees, if you report the ratios of intensities between synuclein in presence of lipids and in absence of lipids, you see that the ratio is about 1 everywhere. So that means you have no interactions between synuclein and your vesicles. But if you increase the temperature, you increase also the lipid fluidity, and you make it possible, so the synuclein starts to interact and bind to the surface of your vesicles. So clearly here you need to record your spectra at 37 degrees. So are we able to do it in cells? And the answer is yes. So here it's just, again, our chimera GFP plus synuclein. We expressed synuclein. So upon doxycycline induction, we changed the medium. We supplemented with C13 and 15 lysines. And well, you see that you can overlay the C-alpha CO correlation spectrum. And we have some peaks that overlay very well. So we miss all the N-terminal lysines because they are too close from this GFP here. But so if you get rid of the GFP now with a different type of labeling, so here we use the precursor of leucine, which is rather cheap, because again, our sample is 10 euros. Well, you see that we have beautiful peaks and good signal to noise in 4 hours. Sorry, I have a problem with okay. Let's go back to the presentation. Yeah, so you see that you have the peaks of the leucine at the C-terminus. Well, they show good intensity, and in the contrary, we have intensity losses at leucine 8 and leucine 38, which correspond to these interactions we observed, we showed before with chaperones. But here, now it's at 37 degrees. So it becomes a little bit more meaningful, in my opinion. We had also the same with the precursor of valines. So you see here all the valines, the valine peaks. but you can notice that the valine 3 is very low in intensity, the same for valine 15. So we have a residue-specific information at 37 degrees again. And recently, we also managed to show that we could have very decent signal to noise at 37 degrees using, sorry, this is wrong here. This is phenylalanine and tyrosine labeling. So we are quite satisfied with this new approach. Once again, because now we are able to measure at 37 degrees, and we are able to actually measure in 4 hours very decent spectra with very decent signal to noise with synuclein expressed at 10 micromolar. The intracellular concentration is 10 micromolar. So we are progressing. So this is what we tested up to now. In gray, you have the labeling that have been shown to be possible by other teams in the literature, and this is what we have shown recently. So what time is it? It's been half an hour that I'm speaking. I don't know, Rams. Do I have time? Or
How many minutes? Do you want 5 minutes? Go ahead.
Yes, well, very quick. So I'm gonna go very quickly through. This is the situation. Just to summarize, this is the scheme. We have cells, and we induce the protein expression. At the same time, we change the medium. We supplement with C13 and 15 amino acids. We put it in a bioreactor, so you see that's how it looks like now in agarose beads or in alginate beads. And we just inject fresh medium constantly to replenish the nutrients. So this is the situation. Very quickly, I wanted to mention that NMR is very powerful to help you with multiple post-translational modifications. Because I was speaking about synuclein quite a lot, but we work also on IDPs. And so I just wanted to mention that IDPs are actually only 1/3 of the human proteome, but they represent 75% of the detected post-translational modifications. And I wanted to highlight the fact that we have developed methods to study these post-translational modifications, especially because statistically, half of the post-translational modifications that are a distance of 4 amino acids or less from the next modification sites, which is very difficult to study using mass spectrometry or Western blots, and using NMR. Well, of course, upon modification, for example, upon phosphorylation, the spectrum before modification in black becomes changes. You have chemical shift perturbations, and you have a new spectrum. So if you record time series of spectra, you will see the black spectrum decreasing, the red spectrum increasing, and you can monitor the chemical reaction using NMR in a residue-specific fashion. And we are also using this method for in-cell NMR studies. I wanted to highlight it because this is, in my opinion, something that people could use more and more for their studies on IDPs and amyloid proteins. And this is true for phosphorylation, but it's virtually true for any kind of post-translational modifications. So these are the PTMs, the modifications that we have studied up to now. With this, I'm gonna stop here. I just want to mention that we are also able to deuterate amino acids, and this deuteration is actually persistent in cells like we see here with deuterated lysines. So it's going to help us to increase the sensitivity for folded proteins and to carry out in-cell NMR studies of folded proteins. This was a quick picture to show you this fact. You see, you have the deuterium shift, and this is all recorded in cell again on this synuclein. Okay, so thank you for your attention. Thank you. So I must mention that Hélène is a PhD student working on, I mean, she's the one who performed all the experiments and all obtained all the results I have shown before. Well, again, thank you for your attention.
Thank you very much, Francois, for a great talk. Let me ask you a very quick question. How do you know that you are able to observe all synuclein populations? Because your ability to observe only monomers using solution MR, maybe you are missing out other populations. Can you?
Yes. So we cannot say that we observe all the synuclein population, all the synuclein molecules. We observe the monomeric soluble disordered population. Now we know how much signal we get from that, so how much signal it represents, and we can just measure the total concentration using Western blots. So of course it's not perfect and not super precise, but we know that we have somehow 10 micromolar. The total amount of synuclein molecules represents something like 10 micromolar. And this is also the amount of NMR signal that we detect.
Great.
So I can't say that, well, maybe there is 10% of lipid binding proteins, I don't know. But
that's great. Yeah. So did you ever observe the population transitioning into some invisible state as a function of time, as a function of other conditions? Did you ever see in your NMR spectra?
So our colleagues, our Swiss colleagues, have shown that when they inhibit the HSP 70, 90, after a while, they see synuclein binding to lipids, and they see the signal decreasing drastically. But they still observe the signal of the C-terminal amino acids, meaning that they observe membrane binding, and actually they had also fluorescent microscopy pictures in agreement with this NMR observation. So it is possible to observe drastic changes through time when you maintain your cells in the spectrometer. I didn't yet, because up to now you've seen that I'm just doing my best to improve the technical aspects. So I didn't try to maintain the cells for a long time and to treat them with oxidative stress or blah blah, etc. So I didn't, but people have had.
Thank you very much. Let's look at the questions from the Q&A. Let's go from the top. There are several questions. Let's go from the question from Nicola Bofinger. Very nice talk. I wanted to ask if doxy induction does not interfere with your alpha-synuclein registry in NMR, since it is known that doxycycline interacts and inhibits alpha-synuclein in vitro and in vivo, based on
Yes.
But
But the concentrations that are likely to inhibit and eventually bind synuclein are very large. I mean, much larger than what we have. Here it's 10 nanogram per milliliter, which is 1,000 times less than what is known to affect synuclein aggregation. So we are way below the concentrations of doxycycline that could affect the behavior of synuclein. The papers showing that you have an impact where you can see clearly that at the concentration that we use, there is no impact. Yeah, nothing happens with synuclein aggregation.
Okay, let's take next question from anonymous attendee. The in-cell signal was big before induction in one of your slides. Why? And is there a way to minimize the signal before induction? Thanks.
It's not the synuclein signal, actually. So maybe I can escape the presentation mode or not. No, I will have to go back. So maybe I guess that the question is about this here, I guess. So it's not the synuclein signal, it's the cellular background signal. Because we maintain the cells during, so we induce the protein expression, and it takes 48 hours. At least our protocol is, so we induce during 48 hours. And during these 48 hours, I mean, the isotope labeled amino acids are integrated in the cellular background, basically. And this is what we see here. A little bit of so you have a little bit of some cellular proteins who integrated these isotope labeled lysines, and so this is with synuclein induction and without synuclein induction. But if you make the subtraction of both, you have the synuclein specific signal. So I guess that was the question.
Okay. Okay, let's take another short question, then we can come back for and revisit other questions. This is one from Ashutosh Tiwari. Very interesting talk. How do you avoid signal interference from other proteins which are abundant E. coli proteins on C13 or nitrogen 15 labeling?
Well, this is I guess it's the same answer. Well, this is not E. coli here. So this is mammalian cells. So I guess the question is about maybe about the previous studies in the past. I don't know.
Sure.
But
if it is
well,
that,
when it's when people look at in cell NMR samples using bacteria, usually the expression of the protein of interest is extremely high, so much higher than anything else. So that's how you manage the problem. Here we just subtract the cellular background, and the cellular background is similar enough with and without induction. It's similar enough, and we manage to have beautiful subtraction and no cellular background signal anymore.
Thank you very much, Francois, for an exciting talk. Let's take the questions at the end. So if we can stay around, it'll be great. The next speaker is Tae Hun Kim from Toronto. He's taking over a position at Case Western. Ray Huang from Guelph is the guest here, and she's going to introduce Tae Hun. Ray, go ahead. Tae Hun, can you share your screen and start the slides?
All right. Hello, everyone. It's my great pleasure to introduce Tae Hun Kim. So Tae Hun, in my opinion, is really a rising star in investigating the structure and the protein-protein interactions between IDPs, in particular those involved in phase separation using NMR, and you probably will see in a number of other biophysical techniques as well. So Tae Hun obtained his undergrad degree in actually pharmaceutical chemistry at the University of Toronto in 2011, and got his PhD also at the University of Toronto under the supervision of Professor Scott Prosser, investigating GPCR actually using fluorine NMR. Afterwards, Tae Hun joined Dr. Julie Foreman-Kay and Louis Kay's lab as a postdoc at U of T and Sick Kids Hospital in Toronto, where he started his scientific journey of looking into IDPs and phase separation. And that's where I had opportunity to overlap with him and got to know him a little bit better as a great young scientist. So in his next step, he's going to move to Case Western Reserve University, I believe in 10 days, to start his independent career as an assistant professor. And definitely exciting journey ahead awaits. So something about Tae Hun's publication record, reading that always gives me a chill. He has 12 publications in total, including 1 on Nature, 1 on Cell, 2 on Science, 3 on JACS, 2 on PNAS, so 1 on eLife, so which kind of summarize about 90% of his papers, and most of them he's either a first author or co-first author. His award list is very long, and I'm probably not going to go through them. So I'm going to highlight the prestigious Banting postdoc fellowship provided by Canadian Institute of Science or Health, and also H.L. Holmes Award for postdoctoral studies from National Research Council of Canada. You don't see that very often in the CV, but he has to decline the Ontario Graduate Scholarship twice and also CIHR Fellowship twice because he had more prestigious awards of fellowship. So I just glad I can give a good shout on Tae Hun, and without further ado, Tae Hun, looking forward to your talk.
Thank you, Ray, for your kind introduction. So thank you for the invitation for today's seminar. Then I'm really looking forward to answer some of the questions if you have one at the end of the seminar. So today I'll be talking about mostly likely the summary of half of my postdoc I've been doing. So which is about looking at the protein-protein interaction within these phase-separated droplets and using NMR technologies. So as Francois talked about, cell is a highly crowded environment. There are a lot of new proteins being synthesized, as well as the new protein, old protein being degraded, and there are a lot of things are happening. And that some of these are possible because, like I said, some of proteins keep getting synthesized, and some of our proteins are being degraded at the same time. But we all agree now that cells are a highly crowded environment. So cell needs to think about or needs to find a way to organize themselves within using in this crowded environment. So one way, if you open up a biology textbook, is that we all know that cell uses a nucleus, ER, Golgi, or the other membrane-bound organelles to organize themselves. But like I mentioned, these cellular organelles shares one common feature, is that they're all lipid-bound membrane organelles. However, there's a new kinds of arising or different types of organelles, such as the membraneless organelles or membraneless biomolecular condensates. Cell uses these condensates to adapt to new environment or translate or react to different stress or different cellular conditions, so that by using these membraneless organelles, and cells can adapt and change its intracellular environment quite dynamically, so that these membraneless organelles are not being accepted or being appreciated or accounted for a lot of cellular functioning that what's happening inside of cell. So it's been discovered by Brandwein and then Tony Hyman, as well as Mike Rosen's lab, that these are these membraneless organelle undergo liquid-liquid phase separation. You can see that under stress condition, these protein labeled with the GFP can undergo these droplet-like formation shape inside of cell, and these can fuse to each other and then have like droplet like behavior. So, however, upon removal of stress, it's just seeing cell needs to adapt to new environment. So, in order to do so, which is not really possible, but if you think about using cell have to use the membrane-bound organelles. So, but because of the fact that cell uses these membraneless organelles, lack of the membrane lipids components is really critical to adapt to this new environment upon removal of stress or adapt to new environment in general. So, my question, especially to this audience, this is quite fascinating because it's been proposed that the single phase or dilute phase or monomeric form of the protein can aggregate potentially within these phase-separated droplets or these membraneless organelle, because of the fact that these proteins tend to be highly concentrated inside the membraneless organelle, such as FUST, EWS, TAP15, TIA1, and many other proteins that are involved in the neurodegenerations are involved, seems to be highly concentrated inside of these membraneless organelles. And when something goes wrong, for example, misfolding can happen inside of these condensates or these membraneless organelle. These can undergo rather irreversible gel-like condensates or even the amyloid fiber. Now, we think it could be responsible or may not be responsible, but we want to know this. So this is possible that these irreversible processes can happen inside these biomolecular condensate. So it is of interest that if we can develop the small molecules or some kind of antibody that can target these or change these membraneless organelle biophysical characteristic of these biophysical characteristic of these membraneless organelle, we can perhaps regulate these processes happening inside these biomolecular condensates. So in my postdoc, I've been studying two proteins that are highly concentrated in the stress granule, neuronal granule, and P bodies, and those are FMRP and caprid-1. These two proteins are, especially FMRP, both proteins are involved in the neurodegeneration as well as the autism spectrum disorder. So in order to so going back to the original question, my interest was to look at the protein-protein interactions happening in these condensates. So I took the reductionist approach. By doing so, I was hoping that I can obtain atomic resolution intermolecular, interprotein-protein intermolecular interaction within these biomolecular condensates. So if you look at the protein sequence of these FMRP caprid protein, in the top panel, using the algorithm developed in the Furman K lab, I was able to see predict which regions are most likely are responsible or driving the phase separation of the entire full-length proteins. And if you look at the FMRP case as well as caprin case, then it's the C-terminal region of the caprin and FMRPs are responsible for driving the phase separation of these proteins. And these C-terminal region of the FMRP and caprins are highly disordered based on these disorder prediction, as well as these C-terminal regions of the proteins are highly positively charged. That it's not surprising that these introduced disorder regions are interacting with the RNA, and these RNA interactions seems to be highly involved in these neurodegenerations. So I became interested. So what I did was I took the C-terminal region of the protein, and I also have to mention that the C-terminal region of the protein I'm highlighting in these two regions are necessary and also they're sufficient to drive the phase separation of the full-length protein. So I took this protein and expressed and purified this protein separately, and then in the addition of the RNA, which is labeled with the green dye, I was able to show that in the presence of RNA, these two proteins can undergo liquid phase separation, as you can see these microscope images. So I was interested in how these proteins can interact with one another. So since they are co-localized inside of a lot of biomolecular membraneless organelles. So I use ITC and to see whether these two proteins can interact with one another. But however, I failed to see any heat changes as I titrate one protein to the other. So, however, what I was able to see is that upon phosphorylation of the FMRP, which is commonly phosphorylate protein, one of the components is also phosphorylate inside of cell, is that I was able to find these phosphorylation-dependent protein-protein interaction happening in using these using this ITC technique. So not only the protein-protein interaction was happening in a phosphorylation-dependent manner, also the phase separation was happening in a phosphorylation-dependent manner. So if you look at the top panel where you have a constant concentration of the phosphorylated FMRP with increasing concentration of fluorophore label of the caprin-1, you can see that as you increase concentration, the lower panel, you fail to see any of these droplet formation. However, the top panel, you can see these nice droplet like structures are forming under microscope. So both protein-protein interactions are happening, as well as the phase separations are happening in a phosphorylation-dependent manner. So then the question is that what are the molecular interactions governing this co-phase separation of this phosphorylated FMRP caprin-1? So in order to answer these questions, I use NMR spectroscopy. So using the carbon detect technology, because I have to work this to do these experiments at the physiological pH 7.4 because of the phosphate charges, then I was able to obtain these beautiful spectrum of the C13 and 15 Caprin-1 in buffer, and then I also acquired the this same type of a spectrum, but however in a condensed phase environment. So now you can see that Caprin now is in a soup of the or sea of the phosphorylated FMRP is displayed in purple, and then double label Caprin now is in the sea of the non-phosphorylated unmodified FMRP in green. So I don't see much of changes in the spectrum chemical shifts. However, what I was able to mostly notably I was able to see is the line broadening in the while comparing these two spectra. So if I were to plot these two chemical shift, these intensity values and normalize with the intensity values by the ones from the dilute phase, I was able to see these plot. And if you look at this, I was able to see these severe peak broadening in this arginine-rich region. As you can see, there are phenylalanine, arginine, and phenylalanine and tyrosines are highly enriched in these aromatic-rich regions. Also, what this means is that these arginine regions are undergoing heterogeneous interaction, a dynamic interaction with the Caprin molecule or with phosphorylated FMRP. So what I was able to see is that these arginine regions are also undergoing severe line broadening. However, if you look at compare the green versus the purple plot, you can see that there are some differences. And if you plot these differences, you can appreciate that the differences are actually present. And not only that, these arginine, based on this data, you can conclude that these arginines are responsible for driving the protein-protein interaction that are interacting with the phosphate of the FMRP. So, so now I know that arginines are important for driving the protein-protein interaction, the phase separations of the interactions. So I mutated all the arginine to lysine and tested this protein-protein interaction as well as the phase separation. So what I was able to see is that I failed to see any protein-protein interaction, nor I couldn't observe any protein a phase separated droplet under microscope, meaning that not only the charges are important because the arginine and lysine have same number of charges, but then what's important also is the pi characteristic at the end of the arginine side chains. So now I've shown that these Caprin protein and the FMRPs, two protein-protein interactions, are also driving the liquid-liquid phase separation in a phosphorylated-independent manner. So I wanted to test whether the other case is true. What I mean by that is now I wanted to test whether the case that Caprin is phosphorylated, and then I added those phosphorylated Caprin to the FMRP. And what I was able to see is that I was able to see these liquid phase separated droplet when the phosphorylation pattern is swapped, meaning that whenever one or the other protein is phosphorylated, you can undergo liquid-liquid phase separation by interacting with the other protein partner. However, when both proteins are phosphorylated, I could not see any droplet forming under a microscope. So then what's the difference between these two droplets? And then since these two proteins are all RNA binding proteins, I decided to add the RNA molecules. So I used SC1 model RNA that we're supposed to recognize or bound by the high tight affinity to the FMRP proteins. So put addition of these RNA molecule into the droplets where the FMRP is phosphorylated, I was able to see some multiphasic droplets behavior where the green label, fluorophore label protein, Caprin-1 is highly concentrated inside of droplet, inner compartment of the droplet, where the phosphorylated FMRP is mostly residing in the outer compartment of the multiphasic droplets. So the question is then, where is the RNA located in the context of these multiphasic droplets? And RNA was highly concentrated in the inner compartment, where the Caprin-1 molecule is highly concentrated in the droplets. So this is a movie, and I was able to confirm that both the outer compartment as well as the inner compartments are undergoing liquid-liquid phase separation, and these two droplets can merge each other, and so have a typical phase, has typical liquid properties, as you can see these under microscope. So then I also decided to add the RNA molecules to the droplets where the Caprin is phosphorylated. instead of FRP being phosphorylated. However, I could not see any multiphasic droplets behavior upon addition of the RNA to this droplet, meaning that the tyrosine could be the key to this multiphasic behavior, because in the case of the bottom panel where caprin is phosphorylated, it was the tyrosine residues that are phosphorylated. So we think those tyrosine that are phosphorylated could be key to these forming these internal droplets. So now going back to these original, so one of the first slides, and I want to talk about these specific organelle, which is mitochondria. So mitochondria has the nickname of the powerhouse of the cellular powerhouse, because it's organelle where a lot of ATP is generated using the electrostatic the gradient of these exists in the mitochondria. So what's been also reported that ATP can have a quite significant role in the modulating phase separation. So shown by the Tony Hyman's lab on the left, you can see that increasing ATP concentration can actually dissolve these phase separated droplets, as well as the shown by the Brangwine at the time he was at the Tony Hyman's lab. He showed that the ATP can change these diffusion or the viscosity of these droplets inside of a cell. So we thought that ATP component aspect of the phase separation could be something really interesting. So this is my summer student, Brandon, adding the ATP magnesium into the caprin-1 molecule, purified caprin-1 solution. And if you look at this, and then you can see that upon addition of ATP, you can see that the solution, purified caprin-1 solution, is being quite turbid. This is often bad for the structural biologists because your purified protein is precipitating. However, when I took this sample under the microscope, you can see that these are not just typical random aggregates you can see in many folded proteins. Rather, that was the case is that there are liquid phase separated droplets under the microscope. Also, what I noticed was quite interesting is that ATP and ADP can induce this phase separation of the caprin-1. However, AMP could not really drive phase separation of the caprin-1 molecule because even at very high concentration of AMP. So we immediately knew that the number of phosphate attached to the nucleotide was quite important for driving the phase separation. So this is the movie showing the liquid behavior of the caprin-1 droplets, and you can see that they are very dynamic and liquid-like properties. So that these are for sure that not phase separated droplet samples. So then question is, where is ATP located in these droplets? So using this fluorophore tag ATP molecule that is depicted at the underneath of the slides, you can see that the TMP ATP is highly concentrated inside of these droplets interacting with the caprin-1. Also, both molecules are highly concentrated in these phase separated droplets. Then how does the ATP drive these phase separation? That's the question, and next question I asked. The question that I try to answer using I go by answering these using these manganese. Manganese is a paramagnetic species, so that so that depending on the distance between the manganese and the protein, my protein NMR signal will change depending on the as the manganese get closer to the protein molecules. So then what I was able to see is that the arginine-rich region at the N terminus and also the C terminals exhibited very relatively lower intensity or volume ratios at these two regions, which is not really surprising because the arginine-rich regions are highly positively charged regions, and as you know, the ATP magnesium or ATP for that matter is our negatively charged moieties. So these are highly charged molecules like to interact with each other because of the opposite charges, so that it's actually driving. So based on this observation, I was able to conclude that the ATP magnesiums are molecules are binding these arginine-rich regions and then neutralizing the positively charged at the arginine-rich regions. So what I think is happening is that the caprin-1, at least in case of caprin-1, there in the absence of ATP magnesium, there's no protein-protein interaction, which is essential part of liquid-liquid phase separation. However, upon addition of the magnesium, which binds these arginine-rich region of the caprin-1, and now these can trigger intermolecular interaction because now, given the fact that these origin-rich regions are highly neutralized in the presence of ATP molecules. So these can undergo liquid-liquid phase separation because of the protein-protein interaction now can happen after these highly positively charged patches are neutralized by the ATP magnesium. So there's no phase separation in the absence of ATP magnesium, and there are a lot of phase separation can happen now in the presence of ATP magnesium. So now the question I wanted to really answer was how these intermolecular interactions are happening even in this condensed phase environment, where the droplet environment. So we use the intermolecular NOE. So I'm not gonna go through all the details which is already in the paper, but what I was able to see is that I was able to detect the intermolecular interaction using this specific NOE method we described in this in my paper. So based on these results, I was able to see that not every single molecule, not every residues are having the same number of contacts or same degree of contact, even in this space separated environment, despite the fact that in the phase separated droplet, the concentration of caprin is roughly around 20 to 30 millimolar. So that one might think that a lot of protein-protein interactions are happening in that where all the residues are interacting with all the residues. However, what we are able to see is that specific residues are recognized as interaction hotspots, whereas some of the residues are not as interaction prone as despite the fact that they are same type of amino acid, for example. So that was very interesting. If I were to plot these intensity or these NOE plots, I was able to identify these interaction hotspots, for example, in these regions, and green highlighted green, these regions highlighted with green, and these regions highlighted with the yellow, and these regions were highlighted with the blue. You can see that the amino acid composition is quite interesting because of GYR is found in the green region, as well as the GYR motif is found in this yellow region. Also, in this blue region, there are a lot of tyrosines and the arginine, also maybe glutamines as well. So that was very interesting. And then we wanted to test that these amino acids are actually important for driving the phase separation. So what we decided to do was we mutated these GRY, or some of these molecules, some of the amino acids in these blue regions are mutated to the alanine, serine, alanine, so that we try to disrupt these intermolecular interaction. And we were able to actually see that by disrupting these, by mutating these GRY or some of the molecules, some of the motif in this blue region to alanine, serine, alanine motifs, we were able to disrupt these or decrease the propensity to phase separate, meaning that indeed these three regions are important for driving the phase separation of the protein and these phase separation of these caprin molecules. So another interesting aspect came from these measuring the simple R2 relaxation rates. So R2 relaxation rates can be interpreted as where the interaction hotspots are happening, because of as the protein-protein interaction happens, these relaxation rates can really go faster. You can think of these high R2 relaxation rates where residue with high relaxation rate, you can think of as where a lot of protein-protein interactions are happening. So not surprisingly, so that you can see that some of the residues are identified using the NOE where actually having these high nitrogen relaxation rate. However, what was really, in my opinion, truly astonishing to me was that you can see the similar type of similar patterns of the R2 relaxation rates happening even in the mixed state or the dilute phase, meaning that for some reason that same kind of same type of interaction that are observed in the same type of interaction observed in the condensed phase was also happening even in the mixed state where there's no phase separation. So that was really interesting because I was able to just multiply certain factors of these lower bottom relaxation rate profile measured in the mixed state or dilute phase, and I was able to get a very similar relaxation profile as the one that observed in the condensed phase environment. So if you compare the NOE plots that I presented before, then you can see that several hotspots were also these two patterns, these profile has a very similar profile, very similar patterns. You can see that some of the residues have high NOE. You can see that some of the residues also have high relaxation rate, meaning that those are actually NOE, these intermolecular NOE, are actually contacts are giving rise to what's responsible for giving rise to high relaxation rates. So I'll skip this slide. Also, so based on this observation is that what we think is happening is that there's a correspondence between the intramolecular interaction happening in the dilute phase or the mixed phase, and whereas also similar type of interactions are replaced with the intermolecular context in the condensed phase. So if I were to describe in this diagram, so there are a lot of extensive intramolecular interactions happening in the dilute phase. However, these interactions can be either satisfied with the intramolecular interaction in the absence of ATP magnesium. However, in the condition where phase separation can occur, these intramolecular interactions, instead of being satisfied by its own chain, these interactions can now satisfy or this free energy is satisfied through the intermolecular interactions. So that was really really interesting observation. I thought was quite noteworthy because of these transition between the intramolecular interaction can be satisfied or converted into to the same degree of the intermolecular interactions. So going back to this, and what I also was able to see is that as I increased the ATP magnesium concentration, initially as measured by these turbidity measurements, you can see the initial turbidity goes up because it's forming these liquid-liquid phase separator droplets. However, as I further increased ATP magnesium, I was able to see the turbidity dropping down. So using NMR spectroscopy, also once again, that I was able to confirm that these ATP magnesiums are now also at these high concentration ATPs are interacting with these arginine-rich region, also at this high 70 millimolar ATP magnesium concentration. Now, even the arginine-rich regions are now also being bound by these ATP magnesium molecules. So with the moderate concentration of ATP magnesium, this can lead to liquid phase separation because of the fact that you can have a charge balance can be achieved using these moderate concentration of ATP magnesium. However, very high concentration under very high concentration ATP magnesium, these no longer can happen because now ATP magnesium now start interacting with this arginine-rich region where now all these heparin ATP magnesium complex is now charge inverted such that there's no longer protein-protein interaction happening. So with this in mind, I think I can explain why there's no phase separation in the absence of ATP magnesium because the charge of the heparin molecule is highly positively charged that can lead to the protein-protein repulsions. However, in the presence of moderate concentration of ATP magnesium, now charge is now balanced and neutralized to some degree that they can actually lead to this favorable protein-protein interaction and lead to these lead to the induction of these liquid phase separated droplets. And in the presence of very high concentration magnesium, also there's charge inversion from positive to neutral and neutral to negative, so that there's no liquid phase separation happening at this high concentration of magnesium. So this was also further verified by my colleague Yuki. Also, he was able to measure the electrostatic potential in these mixed phases as well as the condensed phases, and he was able to come up with these measure these electrostatic potential in these three conditions and confirm that the hypothesis I put forward is actually the truth. So I'll skip this. So I'd like to thank my supervisors, Julie from MK and Louis K. Also, I'd like to thank the key people helped me with the studies, especially Brian Sang, and also other people in the Forman K lab and also Louis K's laboratory. Also, I'd like to thank the funding agency who paid the bills. And with that, I'd like to also, like Ray mentioned, I'm starting my own laboratory at Case Western Reserve University in Cleveland. And I did some self-serving slides and recruiting postdoctoral fellows. I didn't have a chance to talk about my next project, but that also involves the nucleosome core particle, where the Mike Rosen's lab has shown that there's arrays of nucleosome or chromatin can under phase separation. So I have a developing story regarding how DNA damage repairs can be happening in the context of the nucleosome particle. Also, phase separation can be in my future paper, it will show the how phase separation in the context of the DNA damage can be important for facilitating these DNA repair processes. So with that, I'll stop here. I'd like to thank you for your attention and time.
I think Ray is gone, so I'll take over. We can go through the questions in the Q&A. Before that, I have a question for you. So
Yep.
What is so special about intermolecular NOE? What do you have to do to measure them? Do you have anything methodological development? Can you just give some quick details on that?
Yep. So what we did was, in this condensed phase environment, we have two kinds of a caprin molecule. One is the proton C13 and 14 caprin label, which is depicted as a black, and also deuterated C12 and 15 label caprin. So you can imagine that only in case of red, it's only solvent exchangeable protons are present in this red, whereas all the other protons are labeled with the protons here in the black. So we transfer the NOE through filtering, edited filter experiment. For example, we can excite only the aromatic side chains, and then from that we can perform NOE experiment to these deuterated proteins where only the proton is so we transfer NOE from these from the black chain to the red chain, and also and then after that we can just acquire the HSQC spectrum. Since the red chain is the only one that's N15 label, we can get a signal from these red chain. So using these NOE experiments, we can measure these intermolecular NOE, and we can filter out all other intramolecular NOE components.
Do you have accurate distance measurement between the sites?
That's very difficult because the NOE measurements are a mixture of how frequently these contacts are occurring, and as well as how the distance. So it's very difficult to convolve or the distance versus the frequency of these intermolecular NOEs are happening. So, but all I can say is we identify these several hotspots that give rise to these NOEs. But yeah, so what was really surprising to me was that, for example, if you look at the glycine, not all glycines are really having same amount of NOE, meaning that there are register-specific interactions are happening, sequence-specific components are happening in the phase-separated environment. So not only the amino acid type matters, also the sequence matters a lot. So that was really the key message I was able to obtain.
So your arginine to lysine mutation suggested that the arginine is very important for this intermolecular interaction droplet formation.
Yes.
You also
Yes.
mentioned about the pi electrons. Do you think the phenylalanine side chain pi electrons are interacting with the pi electrons from arginine side chain? That's the contact site?
Yeah, so we did exactly the same experiment. So we can, so what we have done is, instead of just exciting just the aromatic side chains, we can also decide to excite just the arginine C deltas, for example, where has a distinct proton chemical shifts. So by doing this, we transfer the arginine from black chain to the red chain or to the amide backbone. Even in that case, we were able to see a lot of NOEs transferred to the side chain as well as the backbone of the red chain. So that was very informative.
Okay, Tae Hun, great talk. Let's go to the Q&A questions. Let's go from the bottom. There's one from anonymous attendee. Is the effect of ATP that you see generalizable?
Yeah, that's a great question. So I don't have data with me at this moment, but I was able to see these phenomena happening with FMRP, the C-terminal region of FMRP, which is also highly positively charged region. Also, another protein I tested is the THXX. I think this is the short 100 amino acid from this helicase domain. So as long as in our hands, as long as the protein or IDRs in terms of these sort of regions are highly enriched with arginine or highly positively charged, these ATP effect, we were able to observe it.
So the next question from the bottom is from Nicholas Fauzi. Can you look at that? It's a long question, so I will read it for you. Do you think that the apparent KD you measure is quantitatively meaningful? The units of micromolar imply it forms a 1-to-1 complex A plus B going to AB, but what is the stoichiometry?
Yeah, that's a great question. So I believe I don't really have a full full comprehensive idea of what's happening. Sorry, with the stoichiometry. So if I were to go back to one of the first slides, these for example, in using the ITC, I can also get the information not just about the KD, also I can get information about stoichiometry. But I don't think we can. This is very accurate because these first of all, these heat measurements is probably a mixture of the protein-protein interaction happening in the dilute phase also, but there's heat involved in the process of phase separation. So the two mixture things are happening. So that's why these KD may not really reflect the KD happening in the dilute phase, but also. So it depends on the charge, in my opinion. I think charge is a big player here. So the phosphorylated FMRP, when it's fully phosphorylated, I think it can have probably close to -10. So so it all depends on the pH, of course. But so yeah, I don't have a full idea about what's happening with this stoichiometry.
Okay, next question from Nicholas Fozzi is, are these proteins phase separating in the ITC? Have you tried changing the concentration and see if you get a different answer? It is impressive you were able to measure the binding by ITC.
Yeah, so I think, like I said, these heat are actually a mixture of the phase separation as well as the protein-protein interaction. So I haven't at least tried to do this ITC measurement at varying concentration of proteins, but yeah, that's actually excellent questions that I should have tried it some time ago.
Okay, the next question in the Q&A is from Sangchul V from Magnet Lab. Are NOEs from intermolecular protein-protein interactions? Answer is yes, right?
Yep.
If there would be some contributions from intramolecular, how can you distinguish them from intermolecular NOEs?
Yeah, this is exactly the reason we use these schemes, so that since we are using the deuterated N15 molecules and the C13 molecules only, but this is no perfect because we have 1% C13 label. Also, we have a very little amount of N15 label, so it's not perfect. But we did a control experiment to show that these are the NOE we're detecting are almost all of them are coming from intermolecular interaction, and very little are coming from intramolecular NOEs. So we are able to accomplish these select for these intermolecular NOE using these selective labeling scheme.
Are you considering a molecular translation or diffusion for intermolecular NOE factors?
So the correlation time is an important factor for the coming to NOE, right? But since these are all in the slow tumbling regimes, so especially in the condensed phase, so I don't think it's a big factor, but we haven't considered the diffusion components of these when it comes to calculating these NOE measurements.
Do you know what is it? Do you know what is the time scale? You said slow. Is it millisecond or even slower? Or maybe
I mean the mixing time.
Your time scale of motion.
Oh, okay. So I believe I have these diffusion constants. So what I can say is at least 100 times faster in the diffusion is 100 times slower in the condensed phase environment. So yeah, so that's, I mean, this is actually quite fast compared to other phase separated samples. That's why we like this caprid molecule, because it gives great spectrum and we can get a lot, and also we can it doesn't gel up. So unlike some of the proteins people have examined, it doesn't form any fiber or amyloid or any of those. So that it's really good for a solution state NMR.
Even if you leave it for a long time, like months, doesn't form any aggregates? I see.
No, it doesn't form any aggregates.
I think we answered Sangchul V's question. Yes, later on clarified this question. I think we addressed that question. The question from anonymous attendee, how can you reliably measure NMR parameters such as R2, transverse relaxation rate, from the condensed phase? Isn't it peak broadening, or overlap
Yeah,
you?
yeah, that's so that's an issue, but we battle it by using many different approaches. So for example, the black spectrum is first of all deuterated. We chose the spectrum sequence, as well as these are acquired of the gigahertz, so that we play this. Also, we optimize the temperatures to have these optimal. This is the spectrum actually acquired from the condensed phase. So given the fact that these are coming from condensed phase, this is excellent quality of spectrum, in my opinion, because others have shown that for example, first, the spectral resolution was not as great as the caprin-1 molecule. So we have to we optimize. Also, pH was low. For example, it's 5.5 to battle against the solvent exchange rate of the amides. So combination of all these things I mentioned was sufficient to lead to this high resolution spectrum, as you can see here.
Great, thank you very much, Tae Hun. Let's go to the questions from Francois. Francois, can you
Yeah,
turn
I'll
on
start
video
sharing.
and audio? Yeah.
I'm here.
Yeah, so let's go to the question that you typed answers. Let's address them so that we can record it. This is from anonymous attendee. Very nice talk. My question is related to the electroporation. So currently you are not adapting this method for protein entry into the cells for your study. Why? Some questions, but not related to this talk. I'll go through them next. So maybe you can answer why you are not using this electroporation method.
Well, this is what I've tried to explain in some slides. Well, basically, electroporation is a delivery method, so that means you need to have high concentrations of the protein you want to study during the delivery step, and this is something that you cannot apply to any protein. Some proteins you cannot have that at millimolar concentrations, basically. So that's one aspect. The second one is, so that means the expression in situ that we perform now is by essence adaptable to a much higher number of proteins. Then we also have access to all the co-translational events that may happen, which is not the case with the delivery methods. Then about the electroporation, questions that this spectator asked. Do you want to?
Let me read it for you. After after electroporation, cell recovery time of cells is 5 hours. Can we increase this time so that we can get desired number of attached hex cells, or can we increase the number of cells or cuvettes?
So the question is related to some specific details from papers 8 years ago. This spectator mentioned, so he's asking a question about the situation that we had to deal with. So when you electroporate your cells in presence of the protein of interest, after the electroporation delivery step, a certain number of cells just die because the treatment is harsh enough to have about half of your cells dying. And the way we managed to maintain, well, to separate the dead cells and the living cells was by just letting them recover on a plate, and the living cells attach to the plate and the dead cells don't. And it takes about 5 hours to really have adherent cells. And so the question is, could you have more cells surviving if you wait more? The answer is, of course, not, because the dead cells don't attach and that's it. Now you can let your cells as much time as you want on your plate, but it won't change the number of surviving cells. And if you want to have more cells, you have to start with more cells and to electroporate more cells, etc., etc.
Okay, the next question is why two continuous steps of electroporation required for the protein entry into the cells?
Yeah, that's again a technical detail, but for those who know, those who work with electroporation and cells, you always have much better results in terms of delivery when you apply two electroporation shocks, because basically the first one destabilizes a little bit the membrane. So during a few milliseconds, you make pores, but the membrane is also destabilized during minutes after that. So when you apply a second shock a few seconds later, the pores that you will create will be even more useful for the delivery, let's say, even though you have a balance. So these pores are maintained a little bit longer, but you will also have a higher death rate using this protocol. So it's a balance to find. You could apply 3 shocks, for example, but
Okay, the next question is from Carmelo La Rosa. This is about the role of free lipids. Free lipids promote lipid interaction with the proteins, peptides and amyloid proteins, IDPs, that enable the entry into membrane from aqueous phase. Do you think it is possible to demonstrate this transfer from aqueous phase to the lipid phase using cells?
I guess it would be it would be feasible if the population, or I mean this phospholipid protein complex would represent a population that is high enough
And fast tumbling.
and it would also depend on the exchange rate, etc. But you would need to have high populations, and I'm not sure that it corresponds to the situation that is evoked here. I mean, that's my feeling, but well, if the population is high enough, maybe yes.
Well, population should be high enough and should be also fast tumbling as well.
Yes.
But the next question from Nicholas Fozzi, I understand that proton N detection is not good. Is proton detected, proton alpha, something that is not as good as C13 direct detected?
So there are different H-alpha detected sequences, NMR sequences. We have published one, actually Andrea Boder has published one, which is very, very efficient last year. And we are in the authors, among the authors. And well, the thing is, all these H-alpha detection methods are, well, they are plagued with the water signal, basically. And you need to have a very good, very good chims. And if your water signal is a little bit broad, which is often the case with cells, as you can guess, you have trouble. So as far, I mean, I have tried, maybe there are things to improve, but up to now, this is not something that has given satisfying results in my hands. And because the peak of the water peak is broad, and you also lose, I mean, quite often with this H-alpha detection, you lose, you have to decide which peaks you lose. So, well, basically, I will not
Okay, very last question.
that, but anyway, the problem is the water peak.
Okay, very last question. You showed a lot of different specific labeling combined with induction. Can you combine them? What happens if you induce with uniform labeling?
Okay, you showed a lot of different specific. Okay. Sorry. we can combine them, and we are doing it now. So we combine so I have shown specific labeling because I wanted to test every one of them. I didn't show you the ones that don't work, by the way. But so we can combine the ones that work now, because they are basically independent in terms of metabolism. Uniform labeling is feasible. It has been shown by our colleagues in Florence, by Enrico Lucinat and his colleagues. It's a little bit more expensive somehow. And you, you control the control of the situation is less perfect, in my opinion. So it's mostly because of costs and because I wanted to have very specific a very specific situation with the cleanest spectra I could. So uniform labeling is feasible. I think that we can reach, we can get a lot of information with the specific labeling also. And basically the thing is, by using specific labeling like we do, we can also include deuterated amino acids. This is what I was mentioning briefly at the end, because and with uniform labeling, it's going to be very expensive to have a uniform deuteration, etc., and the cells will not survive. So that's the reason why. About amidation, I will answer the question to Nick later by mail.
Okay. Thank you both very much. Let me make an announcement. This is the last ZOOMinar for 2022. I would like to thank all the speakers and participants and organizers who helped me to organize this event, these series of events. So we will have a holidays break, and then we'll restart on January 9th with Dr. Per Westermark and another speaker from France. Giving talk, but the schedule is going to be different. It's going to be at 8 o'clock New York time from next year onward, so that people from East can also join. We'll have a number of speakers giving talks from Eastern countries who were missed in the past series. So thank you both very much, Francois and Tae Hun, for your exciting talk, and best wishes and happy holidays to all of you guys.
Yes,
Ciao.
thank
Bye-bye.
you, Rams.